Ever wondered how our bodies actually decide whether we become male or female? It's not as simple as just an X or Y chromosome. In today's video, we are breaking down the fascinating world of reproductive embryology. Let's dive in. For the first 6 weeks inside the womb, the developing embryo looks exactly the same regardless of its genetic sex. The anatomical structures that will become the reproductive system are identical at this stage. After the 6th week, the genetic instructions kick in, and the reproductive organs begin changing into either male or female structures. Sex of the conceptus determined by the sex-related gene, that is SRY gene, present in distal end of the short arm of Y chromosome. If the embryo has a Y chromosome carrying a functioning SRY gene, the indifferentiated gonads are instructed to develop into testes, making the fetus anatomically male. In the absence of a Y chromosome and thus no SRY gene, the default genetic path takes over, and the gonads develop into ovaries, making the fetus anatomically female. Let's clear some anomalies like Turner syndrome and Klinefelter syndrome. Turner syndrome occurs when a baby is born with only one X chromosome instead of two. Its genotype is 45X0. Because there is no Y chromosome, these babies develop as females. Klinefelter syndrome occurs when a male has an extra X chromosome with genotype 47XXY. Because a Y chromosome is present, the SRY gene triggers male development, so these babies are biologically male. This is how the sex of baby is decided. One most important thing is that, for proper development of ovaries, both the X chromosomes should be present, although both the X chromosomes are not needed for presence of ovary. Because of that, individuals with Turner syndrome 45X0 only have one X chromosome, their ovaries fail to develop fully. They degenerate early into non-functional, fibrous tissue known as streak gonads. How does the body actually choose its gender? Let's understand. During early embryonic development, specifically up to the 6th week of gestation, the embryo is in an indifferent stage. This means that whether the embryo is genetically male or female, it physically looks the same. Early in development, a thickening of tissue appears called the genital ridge. This ridge is formed from the intermediate mesoderm and it serves as the physical foundation where ovaries or testes will grow. This physical foundation, the genital ridge, becomes visible under a microscope at around 5 weeks of pregnancy. There are the two pairs of ducts. 1. Wolffian or mesonephric ducts. These are the precursors to the male internal reproductive organs such as the epididymis, vas deferens, and seminal vesicles. 2. Mullerian or parammesonephric ducts. These run alongside the mesonephric ducts and are the precursors to the female internal reproductive organs such as the fallopian tubes, uterus, and upper portion of the vagina. Developmentally, the Wolffian ducts form first. They actually guide and act as a structural template. Without the proper formation and presence of the Wolffian duct, the Mullerian duct cannot grow alongside it and develop properly. By the 9th week of pregnancy, sexual differentiation is actively underway. In males, the testes produce anti-Mullerian hormone, AMH, which causes the Mullerian ducts to regress, while testosterone keeps the Wolffian ducts alive. In females, the absence of AMH and testosterone causes the Wolffian ducts to naturally regress, leaving the Mullerian ducts to develop. By the 10th week, the remaining pair of ducts has fully committed to its path. They begin to actively differentiate into the finalized internal sex organs, either the male tract or the female tract, completing this crucial phase of reproductive system development. Thanks for watching. Subscribe for more updates.